there are a couple of abstracts that were accepted and will be highlighted in this symposium. The first one is by Garrett, a Ph.D. candidate at Georgetown University. Thank you.
It's funny, this could as easily be called more going on with splicing. I was so happy to hear a question about sequencing earlier, but then I realized that this is a very clinically focused crowd. Most of what I'm going to be talking about is about sequencing in Ewing sarcoma. We'll start from the idea of genes, and as you typically see it, it's colored rectangles on an individual line. You have the process of transcription that produces mRNA, the message, the information that's eventually going to be translated and turned to the protein, where the activity happens in the cell. So my part of this is focused on the transcription process, how the DNA gets turned into mRNA, and the individual components of that, namely splicing itself. So if you think about the mRNA, it doesn't exactly replicate the information contained in the gene. Certain elements are removed through splicing. And in fact, there's a segment in between the pre-mRNA where the introns contained in that are taken out, and the exons are what actually turns into the mRNA and produces protein. So if you follow me with that, you can have a series of mRNA messages produced from the same pre-mRNA, and that would give you protein diversity at the end. And that's how people explain the wide range of proteins produced in the relatively small number of genes in the genome. So when we wanted to look at that in Ewing sarcoma, we started with, well, you know, where anyone really starts, you start with your cell lines. And so we took an Ewing sarcoma cell line, TC32 in this case, and one that had knocked down ELIOS-FLI1, and did RNA-seq on these different regions. And you can see, like, we're going to talk about density of RNA in a number of different settings. In this case, this would indicate a splice site because these are units of information that connect these two distal regions. And so you can think of that as information that would indicate the presence of an alternative spliced isoform that doesn't contain that in the mRNA. So what we wanted to do next was look at the entirety of the transcriptome. And RNA-seq itself doesn't do the best job at producing a complete transcriptome because you have fragmentation that goes on and you go through the reconstruction process. De novo reconstruction can be a little dodgy. And so in 2015, we came across a technology called PacBio long-read sequencing. And we essentially can take the entire transcriptome out of a series of cell lines and sequence that to produce a transcriptome that we would use later with the short-read RNA-seq. So we generate this kind of hybrid approach for all of our patient samples. And so when we look at the actual transcripts contained in the transcriptome, you can align it back to the genome. You can see what genes they originated from, that sort of thing. And we found that the genes that we sequenced were largely those that were found in RefSeq and Ensembl, two of the main databases of mRNAs that are used for RNA-seq. However, when you look at the transcripts themselves, the sequence, they didn't match up in the same way. You had a majority coming from some other space of information that wasn't in RefSeq or Ensembl. And so that was astounding to us, very interesting. It suggested that there was a splicing process going on that wasn't common to the databases that existed. So we went to look at what could be generating that difference in the transcriptome, and we found that there was a large degree of retained introns in our transcriptome that weren't found in RefSeq or Ensembl. And so to reference our earlier model would be the contained set of introns that were in our pre-mRNA that weren't getting spliced out properly, or that there was some part of that regulation that wasn't happening as it would have normally. So the next question that you would ask is whether or not that's true in patients, because we've looked at cell lines so far, and that's not, always representative of the patient. So if you take a patient sample, in this case, this was one of Olivier Delatre's patients, and look at our transcriptome, it's across the bottom here, we found a retained intron in our cell line that wasn't in RefSeq or Ensembl, And we aligned our patient sample to it and found that there were, in fact, a decent proportion of reads that align to that retained intron. And so we have strong evidence that those retained introns are present in patients as well. In fact, it's a little bit more complicated than that. You usually have series of retained introns next to each other, but the fact that you have clean splicing near it indicates that something specific is happening with those introns that wouldn't be discovered normally. And in reality, it's kind of messy. There are a lot of retained introns all over the place, which really accounts for that degree of novel information found in the mRNA that we described earlier. So because we're dealing with patients and this retained intron concept, these things are not that closely linked yet, but as early as 2005, people were talking about drugging the spliceosome as a mechanism of treating specific things. It eventually turned into treating cancer and anti-tumor drugs. And in 2015, a paper was published about treating the spliceosome specifically in MYC hyperactivated cells, but they were coming at it from a different direction. They were looking at the spliceosome as a weakness because it produced these retained introns when you partially inhibited it. And that was how you could kill cells. However, we have cells that are retaining introns and staying alive. So we're trying to figure out how that presents a weakness that we can therapeutically target. And so with that information, we know that there's retained introns that seem to be specific to the Ewing sarcoma cells we sequence. And while it's difficult to say whether or not those retained introns are also found in other cancers, we expect to extend that research by looking specifically for those introns now that we've found them in other cell lines. What about the regulation process is a very interesting question. And so we intend to look into the specific splice partners that recognize branch points and actually do the splicing and compose the splicing machinery. We are not sure yet whether or not the degree of intron retention found in these patients is indicative of their response to therapy, but it would be an interesting prognostic marker because it's relatively easy to test. I mean, you can design some primers, induce PCR, and essentially tell the degree of intron retention at specific sites that we identify. The next step is getting into the actual consequence of the spliceosome and whether or not that's a requirement for transformation by EWS-FLI1, which is also something that will be difficult to answer. But as I'm starting my PhD, that's the sort of things that I'll be working on. Rescue Lab, I've been honored to be a part of it since 2012. We did a lot of our PacBio sequencing up at Children's, and the Delattra and Stegmeyer lab provided patient samples. Thank you all.
Sorry. So my background is in epigenetics, DNA methylation. There's an emerging literature that DNA methylation regulates splice site selection, alternative splicing. And there's now some literature, including very recently in Ewing's, that there's methylation heterogeneity in many cancers. And Ewing's, I think, was a recent Nature Medicine paper. Have you thought of looking at epigenetic variability or DNA methylation variability as the driving force of kind of the splicing consequence?
We've certainly thought about it, and there is a reasonable amount of support for the certain histone modifications surrounding exon site selection, and it's something that we'd like to get into. In another arm of research, we're looking at things that modify the epigenome in Ewing sarcoma, and so we hope to be able to tie those things together at some point. It's on our minds.
Just a question. So you're seeing these because you're using the long read sequencing technology. Was that correct? So that you don't see them when you use the small fragments; they just don't map to the reference genome. Is that correct? Or?
That's the idea. More often than not, when you have short reads and you're aligning them to introns, you would account that to noise, or you target it up to something else. I mean, you can deal with things that reads that are spliced across junctions and count those as evidence instead of those that pertain specifically to introns. There are different ways in dealing with that, but we found that the most direct way to sequence introns is to use complete mRNA instead of fragmentation.
And so, I mean, do you feel like you're just capturing intermediates? I mean, you know things are going to splice at different, I mean, so is the technology that's so sensitive, we are catching those intermediates? And, I mean, is this a phenomenon, for instance, this patient, if you took normal tissue cells or something, you don't ever capture these intermediate steps? But.
So, I mean, in part of the protocol of preparation, you do poly-A purification, and so you should theoretically have final processed mRNA that's eventually going to be translated. So hopefully you're getting that. And I mean, I made sure when I was doing the actual processing that what I'm describing is transcript sequence difference comes internally. It's not at the end modifications that might be just degradation or something like that. And it's more than 2 base pairs. So there is good evidence that the mRNA contains different sequence. Yeah, we think it's probably
And you think
real.
that's a cancer-specific effect, like in normal non-transformed cells, is there a way to quantitate, I guess, kind of that? That's a very big bioinformatic question that I have no clue how to do, but.
Yeah, that's a difficult question for Ewing's since we don't really have a cell of origin to compare it to. And so when you look for a transcriptome in different cells, the best way to do that really is to look at a panel of tissue samples that have been RNA-seqed and look at the intron retention in there. And so far, this seems to be something that's occurring in Ewing's sarcoma and not in tissues normally.